Hello everyone, welcome you all in this video tutorial. So in this video lecture, we will discuss about gene therapy. So let's start with the introductory part. So the gene therapy is a method which is used to treat various genetic disorders, and it is a technique which involves the replacement of defective genes in any organism with the healthy ones in order to treat several genetic disorders which are caused by these defective genes. So in this method, the DNA or the deoxyribonucleic acid is introduced into the cell of human body. So basically, the gene therapy is a emerging technique in the field of medicals to treat several genetic disorders which cannot be treated by other conventional methods. So we know that DNA is the nucleic acid which acts as a hereditary material in all the organisms, and DNA consists of genes in its structure, and these genes are the unit for hereditary information, and these genes are responsible for all the cellular activities and inheritance also. So if an offspring inherits defective genes for a particular trait from its parents, then these defective genes also show their functions in its structure. So due to their defective genes, there are several genetic disorders caused in these offsprings. So in the case of gene therapy, this DNA is replaced by, or this defective gene is replaced by the healthy gene in the organism, and by this technique these disorders can be cured. So now let's understand about the history and discovery of the gene therapy. So at first, this concept about the gene therapy was first described by Rogers and Federer, and after some time there were a lot of work done in the field of genetic engineering as well as in gene therapy. So in 1961, scientists named as K. Loriane introduced a functional gene in mammalian cell and see the results. And later in 1971, Carl L. Merrill suggested that DNA insertion could be fixed to fix the mutant genes. So basically, the DNA insertion is also a technique for biotechnology or genetic engineering, where a DNA from a healthy person is inserted into the mutant person or mutant organisms. So Carl Merrill suggested that if the method of DNA insertion is used to cure a mutant gene, then it can be cured. So later on, the first gene therapy was successfully accomplished in the year 1989. So now let's discuss about the types of gene therapy. So basically there are 4 types of gene therapies, which are somatic gene therapy, germline gene therapy, in vivo gene therapy, and ex vivo gene therapy. The first one is somatic gene therapy, in which the somatic cells of human body are used, and in the somatic cells the gene therapy was introduced. So it is performed in a single organism which have the damaged cells, and these cells are replaced by the healthy cells. The second type is germline gene therapy, and this occur in the germline cells or the reproductive cells of the human body. So this method is very common, and it is adopted to prevent and to treat the genetic disease causing variations of the particular genes which are passed from the parent to the offspring and causes different genetic disorders. And by this therapy, the genetic disorders can be cured or prevented in the offspring. Now the third type is in vivo gene therapy. So in the case of in vivo gene therapy, the vector, which is typically a virus, is introduced into the patient, and this achieves a desired biological effect by passing the genetic material. And in the ex vivo gene therapy, instead of a vector, there is a mitotic cells of the particular organism are used. So ex vivo gene therapies are better tolerated and less associated with immune responses. So here this is the diagrammatic presentation for germline gene therapy. So in the germline gene therapy, we know that it occurs in the reproductive cells to prevent the offspring from the genetic disorder. So here in this presentation, we can see a patient couple having dysfunctioning mitochondria in their cells. So they found a donor couple who can donate their mitochondria, and after that the mitochondria of the patient couple were removed, and we have a proper nucleus or a well defined or healthy nuclear DNA which is removed and introduced into the cell of a donor couple which have healthy mitochondria, and thus the offspring have the combination of healthy mitochondria as well as healthy nuclear DNA in its structure. Now, the another type of gene therapy is somatic gene therapy, and the somatic gene therapy can be completed in the somatic cells of our body. So basically this is the diagrammatic presentation. Here we can see a patient having infected lymphocytes or white blood corpuscles. So these infected lymphocytes are removed from its body and a virus inserted with a normal gene. And then this virus infects the lymphocytes with the virus, and there are the normal lymphocytes which are injected in the body. So it needs a vector. In this condition, the virus is acting as a vector for this patient. Now, as we have discussed that gene therapy is a type of genetic engineering, and to introduce gene therapy and to cure diseases with the help of gene therapies, the several products are used to introduce a particular gene or a healthy DNA in the organism. So the gene therapy include two types of vectors, which are viral vectors or non-viral vectors. So the viral vectors are viruses, a particular type of viruses which have plasmids in its structure. So in this order, the virus introduce their genetic material into the host cell, and the retrovirus go a stage further by having their genetic material which copy into the genome of the host cell. The another type of vectors are non-viral vectors. So as the name indicates, they are not viruses, and they present certain advantages over viral methods, because as we have discussed that in the viral vectors, the viruses introduce their genetic material to the host cell, and in this condition there are several chances also that these viruses can infect to these organisms. So the non-vectors are more proficient or more efficient as compared to the viral vectors. Now let's understand about different types of viral vectors which are used as a vector in the process of gene therapy. So there are retroviruses, lentiviruses, adenoviruses and adeno-associated viruses. There are different types of viruses which are used as vectors for the gene therapies. Now the second type are non-viral vectors which do not contain viruses. So there are several types of non-viral vectors which are liposomes, transposons and naked DNA molecules, and all these are used as a vector for the process of gene therapy. Now let's understand about the process of gene therapy and what is the mechanism of gene therapy. So basically in this process, the first step is the selection of the gene of interest. So this is selected by the organism which have a healthy gene for the interest, and after that there is a selection of plasmid which can be of bacterial plasmid or any other plasmid, and then a selection of vectors. So as we have discussed earlier, there are two types of vectors. So the selection of vector done, and after then it is selected that what type of method of gene therapy is used for the insertion of particular gene of interest into the organism. And after that the selection of technique is done, that what technique is used for the gene therapy. So there are several different types of techniques and different types of methods of gene therapy which are used to introduce a gene of interest into a particular cellular organism. Now let's discuss about the applications of gene therapy. So the gene therapy is emerging medical therapy which is used to cure medical diseases or inherited genetic diseases. So in this process the replacement of defective gene with healthy gene occur, and by this process the defective gene can be cured or the effects of defective gene can be limited. The another application is the in vivo gene therapy is used to treat various diseases, and it is also used to treat cancer using transgenes. We know that cancer is nowadays second leading death causing disease in the world, and there is not any proper treatment for the cancer. So the cancer can be treated by the use of transgenes by the method of gene therapy. And there are DNA vaccines also prepared by the help of gene therapies, and different types of inherited diseases like mitochondrial dysfunctioning, diabetes, and many other inherited diseases can be cured by the help of germline gene therapy. So as we have discussed about the applications of gene therapy, instead of all the applications there are very much limitations of these therapies also. The main limitation that this process is very much costly. So no one can afford the gene therapy to cure their diseases. And the second limitation is use of viral vectors. So basically when we use viral vectors to introduce the gene of interest in the host cell, there are chances that viral vectors or the viruses can infect the host cell, which can develop many other diseases in the host cell. And we know that transgenes are a type of vectors or non-viral vectors, so they have very low expression rate. That's why the gene expression rate for these transgenes have very low. And there are several ethical issues for experimentation of gene therapy in live human embryo. So basically in the human embryo, the experimentation of gene therapy is done to cure several genetic disorders, but due to several ethical issues or social issues, it is limited now.